Have you ever wondered what makes a bird a bird? Or what makes a monkey a monkey? These are just some very basic questions that having the complete DNA sequence, the genome of a species, can tell us. It's like having the blueprint to its life. We in the genomics community have seen an exponential increase in the number of species that have genomes available publicly. What this means is for over 5,000 species, we now have that blueprint. There is even a project that aims to sequence the genomes of 70,000 living and endangered species, creating something called Genome Ark, kind of like Noah's Ark, but with the genomes instead of the actual animals in it. And with this information, not only can we answer those very basic questions, but now we have the ability to record and preserve their genetic information before they go extinct, and in some cases, even be able to use this information in conservation efforts. But just having the genomic sequence of a species does not really mean much all by itself. In our lab, we are very interested in developing methods that can help us contextualize and understand the contents of these genome assemblies. I work on methods to annotate genomes. Genome annotation, to put it very simply, is a process where you take in the DNA sequence of a species and then map the correct genes to the correct location, giving the genome meaning. Think of it as you have a new language, the language of genomics. The A's, C's, G's, and T's, the building blocks of the DNA, are like the letters. Segments of the DNA are like words. So now you know what all the words are, but you don't know what they mean. So how would you communicate? Gene annotation is the process where we confer meaning to these words. Now, biologists have context to work with these sequences. They can actually use them in their research. My software, the Comparative Annotation Toolkit, works on doing exactly that. It makes scientists who study certain model organisms or certain species be able to annotate genes on their assemblies by themselves. And what that means is that now they can study these genes, the proteins that these genes make, and then use that to discover novel biological insights. What insights, you might ask? Well, for example, with my collaborators, I'm using gene annotation techniques to study and try to understand the evolutionary processes of a set of genes in us and our closest primate relatives to try and make sense of what makes our brains different than, say, a chimpanzee's. And this is just the beginning. There's so much biology to be discovered, and I'm so excited for it all. Thank you.